This presentation is about mites in hemp. My co-authors on this presentation are Dr. Cindy McKenzie from USDA ARS Lab in Fort Pierce and Dr. Juanita Pompano, the County Extension Specialist in Lake County. Mites are key pests in many crops, including hemp. These pests can attack plants both in the greenhouse and the field. They can cause defoliation, distorted growth, and even kill plants. Spider mites are known to disrupt the water balance in certain crops. We'll briefly talk about Eriophyidae, Tarsonemidae, and Tetranychidae mites, which are the three most important groups of mites attacking hemp in Florida. Integrated Pest Management for Floriculture and Nurseries by the University of California is an excellent primer for developing IPM programs for ornamentals and, in my estimation, for hemp as well. It can be purchased from the University of California or through Amazon. Physical and cultural control. Remove weeds in and around your production area. Dispose of plant refuse. Limit access to crops by pests, making sure that the walls and screens and doors are all intact. Limit all plant material other than your crop in the greenhouse. Caution workers that mites can hitchhike on people as well as other plant materials that are brought into the greenhouse. Quarantine any new plant materials. You have too much to lose by bringing in contaminated plant material and infesting your main crop. Scouting is the foundation of any pest management program. It's absolutely critical. Most growers try to have someone scout their crops at least once a week. The University of Florida, Mid-Florida Research and Education Center in Apopka has an annual training program to teach new scouts the process. Because the mite species that attack hemp are so small, you will need a good hand lens. I recommend having both a 10x and a 20x hand lens. I prefer the type shown in this photo. A good hand lens can be obtained from many different sources and might cost you as much as $50, but they are worth the investment. The ones shown in this photo are Bosch & Lomb Coddington 10x and 20x. Eriophyidae. The Eriophyidae contain such mites as the hemp russet mite and the tomato russet mite. The biology of hemp russet mite hasn't been studied in detail. We refer to what is known about a closely related species, the tomato russet mite, to gain some insight into what the biology of this particular pest might be. The tomato russet mite prefers conditions between 70 and 80 degrees Fahrenheit and 30% relative humidity. Generation time is about a week, and female lives several weeks and lays 10 to 53 eggs. Because hemp russet mite and broad mites are so small, you generally detect the damage before you detect the mites. Symptoms of these mites feeding can be variable and thus difficult to attribute to a specific species or cause. I scout for abnormal growth and or coloration of the leaves. We often see leaves that become curled or distorted. The leaf edges may become curled or folded toward the center on the top of the leaf. The color might appear dull and possibly yellow or bronze. Once you detect abnormal foliage, a sample should be collected and viewed under at least a 20x hand lens or a dissecting microscope to determine if the leaf is infested with either russet mite or broad mites. When using a hand lens, it is recommended that you hold the leaf so that the sun or other bright light is fully illuminating the leaf surface. View the leaf in a way such that the light is unobstructed. If infested, the light will reflect off the mites and you will probably notice some movement. Russet mites are first noted on the tops of the leaves, although the bottoms may be infested as well, but they're easier to see on the top, while broad mites will initially inhabit the bottoms. I have read that others feel that broad mites inhabit the tops first, but that has not been my experience. As the populations grow, all portions of the leaves and stems or flowers become infested. This is simply a close-up of a portion of the leaf in the previous slide. You can see the top of the leaf is covered with Eriophyidae mites, and you can see the edges are beginning to sort of curl over toward the center line of the dorsal surface of the leaf. This is even a closer view where you can see the mites moving along the leaf surface. The Tarsonemidae contain the broad mites, which are about twice the size of an Eriophyidae mite. There are a number of different Tarsonemidae mites that might infest the leaf surface. Most of them just feed on detritus, dead insects, fungi, those types of things, whereas the broad mite is a significant pest. You can tell the broad mite or that you have broad mites by a couple characters. First of all, the eggs have little tiny white facets all along in parallel rows on all surfaces. Secondly, the adult female may have a white stripe that's visible along the dorsal surface from the tail up to the head. In this slide you can see an immature broad mite female. You can tell it's immature because it doesn't have the white stripe on its back. And then 3 of the mite eggs with the facets. This is a pair of broad mites that are about to mate. The male will carry the female around with the tip of its abdomen. In this slide you can see an adult female coming in from the top and going along the vein. You can see a number of eggs, and you'll quickly see a mating pair, or a pair about ready to mate, with the male carrying the female. How do broad mites get into the greenhouse? Broad mites can hitchhike on people's clothing. They can hitchhike on other insects, especially on whiteflies, as seen in the top 2 photos in this slide, where the broad mites have attached themselves to the legs of an adult whitefly. They also will attach themselves to legs of aphids. We often recommend to growers, if you find aphids or whiteflies in your nursery, you should begin to scout a little bit better for broad mites. They can also be brought in by air currents through breaches in the screening. They can move from plant to plant just by crawling. And then they gain entrance in many cases on infested transplants. This is the same with Eriophyidae mites as well. We often see mites on transplants and thus gain entry into your production area. The symptoms shown in these 4 slides are somewhat similar to what you might see if they were infested with Eriophyidae mites. But in this particular case, all these plants were infested with broad mites. If you look closely, you can see that some of the leaves are beginning to curl over at the tip. Along the edges, you start to see them, the edges curl up, such as in the second photo on the right upper row. And in some cases you'll notice that the leaf surface is not smooth. It will start to pucker and get bumps along it, and this is also a sign that you might have broad mites. You can see also that the leaves are starting to curl inwards toward the midrib. And sometimes, like in the bottom right, you will see some discoloration, and also you will see a leaf almost dead center where the edges are beginning to curl over. Tetranychidae, or spider mites, contain a number of different species that attack plants. Two most important in Florida and on hemp would be the two-spotted spider mite, Tetranychus urticae, and then the glover mite, which would be Tetranychus gloveri. In this slide, you can see a link to a website that has the biology in great detail for two-spotted spider mite, and then it has a chapter or so on the biology of Phytocoelus persimilis, a biological control agent for managing spider mites. In this slide, you can see various quiescent stages and eggs of the two-spotted spider mite. When you see the legs sticking straight out in front of it or behind it and kind of knuckled over, they're in this quiescent stage or resting stage. They're in the process of getting ready to molt to the next active stage. They don't move, and sometimes people mistake them for being dead. And so you can see the legs in these mites are kind of sticking out in front and knuckled over. In this slide, you see a male and female two-spotted spider mite, and then the eggs. The male has kind of a pointed abdomen and is much smaller than the female, that's more robust, and generally you can see the sides are dark in color, which results from their feeding on the plant cells. In this slide, you can see Tetranychus gloveri, which is the other species of spider mite I mentioned, as being a major problem in Florida. It's a different color than two-spotted spider mite. It's not a red spider mite, which is sometimes, or Cinnabarinus, but it is a totally different species called Tetranychus gloveri. It gets this kind of brick red or color. They can sometimes be a brighter red. The legs can be a very light to almost white color, and in this case they're taking on a little bit of a darker coloration to orange in color. The damage that these mites cause are very similar. In this case, it's damage caused by Tetranychus gloveri or glover mite. They poke individual cells and remove the contents, thus killing the cell. You get yellow stippling as the cells are killed, and the more damage that the leaf incurs, the greater the discoloration and the larger the yellow patches become. As the leaf starts to deteriorate and is not a very good food source for the mites will begin to web as a mechanism for moving from one plant to another to find another suitable feeding site. Chemical control of mites in hemp. Chemical control can be somewhat problematic. This is a brand new crop, even though it's been around for hundreds of years. We haven't been able to do much research on developing control tactics. Okay, so we have a limited availability of pesticides to use on hemp. Data is hard to find on efficacy and plant safety for those materials that are registered. We don't know much about the impact on biological control agents from some of these compounds. We can glean some of the information where these materials are used in other crops and have been tested on natural enemies, but we really don't have a lot of data on what's safe to use in an IPM program. Customers can be reluctant to buy non-organic crops or products that have been contaminated with anything, including soaps and oils, so it limits the arsenal that we have in terms of chemicals to manage mites. This is a photo of the first page of a document produced by the Florida Department of Agriculture and Consumer Services. It's a guide list to the pesticide products that they've decided are legal to use on hemp in the state of Florida. There are a couple categories that you need to pay a little bit of attention to. There are some that have the heading under EPA registration, FIFRA 25B, which is the Federal Insecticide, Fungicide, and Rodenticide Act. It is a designation that the chemical products containing active and inert ingredients considered minimal risk and thus are registered under the Section 25B designation of the Federal Insecticide Rodenticide Act. Okay. Now the other ones have a number, which means that they've gone through a different process, full registration for either hemp or other crops, and they are considered legal to use. Now they have to, the registrant or the company that makes the material have to submit a more detailed data package to EPA to obtain a review as to their safety and applicability for a particular crop. In this case, the state of Florida read the label and decided that it has the appropriate verbiage on the label to allow it to be used on hemp. Okay, so you can see that there's various azadirachtin made by different companies with full EPA registrations. Now, these packages are not required to have efficacy and phytotoxicity data, but generally there are data for these types of compounds available to show just how effective they are on certain pests, and they also contain some information as to how to use them safely and on a particular crop, such as hemp, a high valued crop, so you need to pay attention to phytotoxicity. Because many of the chemicals that are in that particular list are listed as 25B, you need to probably use a few criteria to select which ones of them you might use. One would be availability. Okay, some of these may not be available in local agricultural products for sales or retail outlet. You may have to order them online and have them shipped to you. Some of these materials may not be sold in useful quantities. In some cases they may be in small bottles, 8-ounce containers, and if you're trying to treat a large acreage, they wouldn't be appropriate. You may need to search online to see if the material that you would like to use comes in a suitable quantity. And you really need to see if there's any information out there on plant safety and efficacy. We always recommend that growers conduct their own phytotoxicity trials. In other words, you can have a few plants that you don't spray anything on, and then you would have 5-10 plants that you would spray the maximum labeled rate that you might use in the greenhouse or in the field on those plants. And I would repeat a number of times, say weekly for about 3 weeks. Just kind of mimic what you might envision using the product and how you might envision using the product, and then evaluate them compared to the ones that were not treated. So you'll get an idea of how safe the materials are to spray on hemp under your particular conditions. When it comes to using oils on your crop, we recommend that you use the low-end label rates first. So try low rates to see if it's safe on your crop. We recommend to apply when it's not too hot and when there's good conditions for drying. We also recommend that you pay very close attention to any kind of label warnings that might be present. Some labels will warn you that using oils in situations where the plants might be treated with sulfur or have been treated with a sulfur-containing compound, that you can run into phytotoxicity issues. There are other interactions that might be present, so again, look at your label and pay very close attention to any warnings. There are 3 compounds that we have tested on hemp or others have tested for phytotoxicity and efficacy from the list that DPI or the FDACs put on the bulletin or the document that I mentioned earlier. One is sulfoyl X that has a full EPA registration. There's tetracurb, which we've tested on whitefly, and we will be looking at the hemp russet mite and broad mites here shortly. That is a FIFRA 25B. We know that it's safe to the hemp plant, but we don't know about efficacy on mites at the present time. And then there's golden pest spray oil that's active on a number of different species and pests, including mites. Biological control of mites in hemp is another tool that you can use for managing mite, and there are a number of growers reporting successful control with certain predatory mites, especially for two-spotted spider mite and broad mites. Hemp mite programs are successful. Biological control of Eriophyidae mites. The three predators named in this slide are listed on the document given below, Guidelines for Purchasing and Using Commercial Natural Enemies in North America, with a link to where it can be downloaded from, that list these predators as being predators of russet mites, probably tomato russet mite. We currently don't have any data to support the use of these predators for hemp russet mite, but we have a project that's been funded that we will evaluate them on hemp russet mite in greenhouses in the state of Florida. Biological control of Tarsonemidae mites, however, in both ornamentals, vegetables, and in hemp has been successful by using at least one of the predators given on this slide, Amblysseus swirsky, Amblysseus cucumeris, and Neosuilus californicus. We've had good success at the University of Florida Research Center in Apopka managing broad mites on hemp using swirsky and californicus. Others have claims that they have had success with cucumeris. This is a video of Amblysseus swirsky feeding on a thrips. Now, the reason for showing this video is that I could say it was Amblysseus cucumeris or possibly Neosuilus californicus or a couple other different species of predatory mites that are commercially available. You really can't tell the difference between these mites by using your naked eye. You would have to mount them and send them off to a specialist. The four predators in this slide are all sold commercially. They are all known to be good predators of spider mites. Phytocoelus persimilis is, however, a specialist predator for spider mites. It will starve to death before it will feed on a broad mite or an Eriophyidae mite, so it is used almost exclusively for controlling spider mites. If you have issues with broad mites or Eriophyidae, hemp russet mite, tomato russet mite, you will probably need to release a different predator in conjunction with Phytocoelus persimilis. My favorite predator that we use a lot in ornamentals is Neosuilus californicus. It'll feed on the major mites that we have to deal with, including two-spotted spider mite and broad mite. Neosuilus fallacius we don't use much in Florida. It is a good predator. It's pesticide resistant in some colonies. It is able to withstand colder climates, and so it might be good for use in hemp in northern parts of Florida. Feltiella is also an excellent spider mite predator. There is a link that I mentioned earlier about two-spotted spider mites on my website that goes into great detail on spider mites, and then there's another link on that site on information on the biology of Phytocoelus persimilis, which is a predator of spider mites mainly. It will not feed on broad mites, so if you are growing hemp, you'll have to release something in addition to Phytocoelus persimilis to control the broad mites. In this case, you can see a dead two-spotted spider mite. In this video, please note the shape of this Phytocoelus persimilis female's body. It's kind of rotund, swollen relative to the other predators that you might see, such as Californicus or Swirsky. This species is sold commercially and feeds on and is very effective at controlling spider mites on hemp. In this slide, you see a comparison of the predatory mite egg at the bottom and a two-spotted spider mite egg up in the right-hand corner. Now, the spider mite egg is about half the size of the predator, and it is spherical in shape. This predatory mite is more oblong. It almost looks like two spider mite eggs stuck end to end. Now, this is a little bit of an end-on view of it, but it's a somewhat longer and side view. You need to know how to detect the difference between these eggs, because sometimes the only indication that the predators are established in your crop is that you'll start to see predatory mite eggs. It's also an important factor to, once you release them, to try to find these eggs to demonstrate to you that the predators have truly established in your crop. This predatory mite is a good choice when the mite populations are relatively low, because it doesn't need as many prey to maintain itself and a reproducing population compared to Phytocoelus persimilis. Neocoelus californicus will feed on more than just spider mites. I prefer using californicus, more effective than persimilis at maintaining populations at low levels. It needs fewer eggs per day to maintain oviposition, and it can feed on alternate prey, including broad mites and other small arthropods. The species is more tolerant of pesticides than persimilis is as well. This is a slide of the Feltiella predatory gall midge. In this case, it's a larva feeding on spider mites. You can see the small egg here at the bottom of the slide. You can see a larger one here, and this is a full-grown larva with some dead mites laying around as well. This is the larval stage of the predatory gall midge Feltiella attacking a spider mite. The gall midge uses its mouth parts to penetrate, in this case, the leg of a spider mite, and it uses its leg, the mite's leg, basically as a straw, from which it will remove the fluids from the mite body. Common reasons for biological control programs failing. Education is critical. You need to know what the prey mites look like as well as the predatory mites. Knowing what the various eggs look like is also extremely helpful. Often, predatory mite eggs may be the only sign that the predators are present on your plants and reproducing. Secondly, there are a number of Tarsonemidae mites that feed on pollen, decaying plant material, and fungi that can be found on plants. These are not pests. There is no need to treat for them. So being able to identify the pest species, such as broad mite eggs, is important. Educating yourself on how to release natural enemies, the biology of the mites, what to expect, and other important issues will help prevent failures. Goals poorly defined. Setting realistic goals, especially in the beginning, helps you determine if you are succeeding or failing, and if this is a control tactic you wish to proceed with. Starting on a small group of plants will allow you to learn what to expect so that you can set realistic goals for establishing a program on a larger scale. No plan. Having a plan as to where you can get help, natural enemies, and other information is also important. You will need to decide on when and how you will release predators. Scheduling predatory mite deliveries so that they are in your hands when they are needed is also an important consideration. Plan to have pesticide-free cuttings is also needed, and it should be planned for as well. Patience. There's a learning curve with using biological control. Natural enemies take time to become established, and begin to exert control of the plant pests. Pesticide residues. Pesticide residues can result in total failures. This is a common issue in production of ornamental plants. Predatory mites are often more sensitive to pesticides than the pests. Poor communication. Everyone involved in the production of the crop must be on the same page with regards to the goals, expectations, schedule, and management of the crop. This includes the office team that might be in charge of ordering natural enemies and receiving the shipments. They need to know when the product is going to be delivered. This is a living product that is perishable. The predators shouldn't sit in the sun or in the receiving area for a long period of time. Quality of natural enemies. Examine a sample of your shipment as soon as you can. The company that produces predatory mites may ship a high quality and viable product, but a lot of negative things can happen during shipping. The most critical being the product is exposed to extremes of temperature that can render the product useless. Don't just look to see if the predators are alive. It is highly recommended that you find a mite-infested plant and release some predators on that plant. Return to the plant periodically over the next few days to monitor and the establishment and reproduction of the predators. We expect the predators to establish and begin to lay eggs. Unrealistic expectations. Biological control is only one tool in your toolbox. Don't expect them to control your mite problems quicker than pesticides. They can outperform pesticides over time, but you can't expect them to cure a rampant pest population. There are a number of links that were mentioned during this talk. There are 5 listed on this particular slide. We will also add links to the home page listed in number 5 below, MREC, IFAS, UFL, edu/hemp, insects, and mites. And this can be referred to in the future for additional links that we feel are important and for the management of mites in hemp. This slide contains my contact information, my phone number, my email, and a link to my main website. This slide contains the information for Dr. Cindy McKenzie. This slide contains the contact information for Dr. Juanita Pompano, and I want to thank everybody for viewing this material.